Assalamualaikum everyone. Okay, Assalamualaikum everyone. Hope you're having a good finals week. Today we're gonna do MOL 1 to 5 practice questions. I'm Libaan and I'm doing it with my colleague Ashaz. Ashaz, you want to introduce yourself?
Yeah, 1 second. I'm Syed. I'm a second year medical student, and I'll be giving the latter part of this PAL session. And my colleague Libaan will give the first part.
Yeah, so we're gonna get into the questions now, and you guys can write the answers in the chat box. I'll be seeing into it. Okay, so first question. Which of the following best explains how Bordetella petrosis leads to increased mucus production in the lungs? First option, it activates G-alpha S proteins, leading to increased AC activity and elevated CAMP levels, or it directly activates AC, causing excess CAMP production, or it inhibits G-alpha inhibitory protein by locking it in GDP-bound state, and D, it stimulates phosphodiesterase, decreasing CAMP. So, anyone knows the answer? Okay, I'll tell you the answer. It's C, it inhibits G-alpha inhibitory protein by locking it in the GDP-bound state because petrosis, which is also called as whooping cough, impairs and inhibits the G-alpha inhibitory subunit of the whole GPCR thing. And it binds in lungs and it binds for a very long time. And now that G-alpha I, normally it would inhibit AC. And AC, if it's not inhibited, it's going to increase CAMP, and because of the increased CAMP, there's going to be overproduction of mucus. So basically, the whole thing is like inhibiting the inhibitor. You're inhibiting the G-alpha I, and therefore we have overproduction of mucus. Moving on. Question 2. Cholera toxin causes watery diarrhea by A, inhibiting AC, reducing CAMP levels, or permanently activating G-alpha S, increasing CAMP, or blocking the CFTR channels in the intestinal cell, or degrading the insulin protein via SH2 domains. I don't see any answers, so I'm going to answer this. It's permanently activating G-alpha S, increasing CAMP level, because cholera toxin impairs something known as the GAP enzyme. And what the GAP enzyme does is it hydrolyzes GTP into GDP. And GTP is guanine triphosphate. It hydrolyzes it to guanine diphosphate. Now there's increased adenylate cyclase activity. And what did I tell you about AC? If AC increases, CAMP increases. And if CAMP increases, the channels are going to stay open for longer. Like the CFTR channel is going to be active and it's going to be open for longer, and therefore chloride is going to go in and it causes watery diarrhea in the lungs, and the lungs fill up. So it's basically hydrolysis of GDP, which is the active state, to the inactive state via the GAP enzyme. And this is from your first lecture. And when I looked at the first lecture, you guys have like questions at the end of your lecture, and I heard from some of your colleagues that the doctor usually just rewords these questions and gives it to your final. So I feel like these two concepts are important, so focus on them. And the next question is about GPCRs. So which of the following statements about GPCRs is true? They are found in both prokaryotic and eukaryotic organisms. The intracellular N-terminal region of GPCRs binds to the ligand, or the GPCRs consist of 5 transmembrane alpha helices, or the GPCRs interact with the heterotrimeric G protein complex via their C-terminal region. I should see some answers on this because it's an easy question. Okay, so the answer is GPCRs interact with heterotrimeric G protein complexes via their C-terminal region. And this is directly from your slides. Know the slide well, because GPCRs is actually a high yield concept. So first things, they are known as the 7 transmembrane. That's why the option C was wrong. They don't have 5 transmembrane helices, they have 7. And also the N-terminal region binds to the ligand in the extracellular domain. The option over here said the intracellular. It's supposed to be extracellular domain. And that's all you need to know about GPCRs. Moving on. The protein insulin receptor substrate 1, IRS1, is phosphorylated at the tyrosine residue by which enzyme? Protein kinase A, protein kinase B, protein kinase C, or tyrosine kinase by PI3K, or tyrosine kinase by insulin receptor. Any answers? Okay, guys should start answering. Okay, so it's tyrosine kinase by insulin receptor. I'll tell you why. So this is the diagram directly from your slides. You see, so the insulin receptor is actually a tyrosine kinase receptor. And it has two parts, an alpha part and a beta part. And the beta part has these two tyrosine hanging from it. And what happens is when the insulin comes and binds to its receptor, it gets activated. And when it gets activated, it's going to autophosphorylate. That means on its own, the beta subunits have something called as the tyrosine kinase enzyme. Now, these are going to phosphorylate its own tyrosine. And then that phosphorylated tyrosine on this receptor would go on to phosphorylate other receptors, like you see here, the other receptor. It's phosphorylating the other IRS tyrosine phosphate. So that was what the question was about. The protein insulin receptor substrate is phosphorylated at by which enzyme? It's the tyrosine kinase by the insulin receptor itself. Okay, moving on. The SH2 domain and insulin signaling primarily binds to phosphorylated tyrosine residues or hydrolyzes GTP to terminate RAS, or acts as a steroid hormone receptor in the nucleus, or directly phosphorylates GLUT4 transporters. So the answer is A, binds to phosphorylated tyrosine residues on IRS1. And this is just something you guys should know, that the SH2 domain binds to phosphotyrosines on IRS1 to recruit proteins like PI3K, initiating downstream signaling. I think you can see it here in this diagram that it goes on to activate multiple pathways, out of which one of them is the PI3K pathway. And that's how it's going to do like glucose metabolism and stuff. This is just something you guys should know. And next question. Okay. Nuclear initiated steroid signaling differs from membrane-initiated steroid signaling in that it's just asking how different, why they differ. So option A activates GPCRs to rapidly increase calcium levels, or B binds to intracellular receptors to regulate gene transcription, or C requires secondary messengers like CAMP or IP3, or D phosphorylates STAT proteins for immediate response. And the answer is B, it binds to intracellular receptors to regulate gene transcription. Well, it's actually in the name itself, guys. NIS is like nuclear receptors, and MIS is like membrane receptors. So nuclear is inside, it's like in the center of the cell. You can see it here. It crossed the estrogens, cross the plasma membrane. They go through, they travel all the way to the nuclear membrane, and that's where all of things happen, like the DNA transcription and stuff happen. So therefore, it's going to be binds intracellular receptors. And that's it. Next question. Which of the following sequences can most likely be recognized by restriction enzyme? I'll give you a minute to see the options. Okay, so the answer is A, because it's a palindrome. So sequence A is a palindrome, and most restriction sites are palindromes. Now, restriction endonucleases can only act on sites which are palindrome, and restriction endonucleases are like molecular scissors. They come and they snip your DNA where they find a palindrome. So AAGCTT would, the palindrome would be, it could be, read both ways. When you read it from the left to right, or you read it from the right to left, it's going to be the same. So that was that. And now, which of the following techniques is most appropriate for analyzing gene expression differences between two cell types by converting mRNA into cDNA? So I think this is pretty easy as well. It's reverse transcriptase. What else is going to convert mRNA into complementary DNA? It has to be reverse transcriptase. And this is an important concept, guys, so make sure you know it. It's used in techniques like microarrays and RT-PCR, which is literally reverse transcriptase polymerase chain reaction. So know that well. Restriction restriction endonuclease and reverse transcriptase are two very important enzymes that you guys can get tested on, and I'm expecting a question on either of these. And next question, an oncologist would like to determine the global patterns of gene expression in two different types of tumor cells in order to develop the most appropriate form of chemotherapy for each patient. Which of the following techniques would be the most appropriate for this purpose? Southern blot, Western blot, microarray, ELISA, or northern blot? It has to be microarray. It has to be microarray because the question said an oncologist and the question said tumor cells. So if you guys see anything about cancer, most likely the option is going to be microarray, and I'll tell you why. So microarrays, which is also called a DNA chip or a gene chip, this is the thing, the square with the grids or panels or the circles. It's a lab tool used to detect expression of thousands of genes at once. And it does this by, actually, the main concept of microarray is to detect SNPs. And what happens in tumor cells is we have a single nucleotide polymorphism. That means a single, a single nucleotide would be mutated. If it's supposed to be glycine, it could become valine. And if it detects such minute details. I'll tell you how. If you see, this is directly from your slides again, the diagram. And this, we take a normal cell mRNA and we take a cancer cell mRNA. Okay, and suppose I labeled the normal cell green and I labeled the cancer cell red. Labeling is basically giving it color so we know what is what. And then I ran the mRNA to a reverse transcriptase and I made it a cDNA. Now I need to make it a cDNA because I need to get something called as a probe. And we get the probes from DNA itself. So a probe is like a small fragment of what we want to test. And then we mix these both and we hybridize to the DNA microarray. That means in this chip itself, I have, in these circles, I have the thing that I want to find. So I put these on the chip, and if I get a red spot, that means the cancer cell is producing its message because I labeled it red. And if I get a green spot, that means it's the normal part because I labeled the normal cell green. And if I get the black dot, that means, and if I get a black dot, neither of the cell produces their message. And if it's a yellow spot, it's going to be like the both cells produce the same amount of message. So that's the concept of microarray. And you can expect a question on this. Moving on. Okay, which technique detects specific proteins? Very easy, very easy. I'm sure you guys know this by now. It's Western blot. So please, please, please remember that Western blot is for proteins, Northern blot is for RNA, and Southern blot is for DNA. And it's a sure question. You will either get a question on either of these. There will definitely be one of these three blots in the question. There's no MOL 1 to 5 without this question. So please remember this. And don't get confused between Northern and Southern because people tend to do it. Northern is for RNA and Southern is for DNA. Okay, next. What test is used for pre-implantation genetic diagnosis of fetuses? A, PCR, B, Southern blot, C, Northern blot, or D, Western blot? And this is also pretty easy. And directly from slides, it's PCR. And I'll tell you why it's PCR. So we're doing a pre-implantation genetic diagnosis. That means before we are doing this test on the egg, before, like, if you're doing an IVF and there's an egg outside of the offside of the body, and we're trying to test on that thing. Why we do PCR is because we can't take much of the sample, and in cases where we can't take much of the sample, we take a little bit and we run it in PCR. And the whole concept of PCR is to amplify. Therefore, it's typically performed in pre-implantation genetic diagnosis, and it allows for identification of genetic mutations before implantation during IVF. And in your question, you might see like even a diagram of an egg. It's like a purple diagram. I think it was on your slides as well. I forgot to add it over here. And you'll see an egg, and you will see like this syringe trying to, trying to do the implantation in the lab. And they'll ask you what kind of, what kind of test would you do in this case. They might not tell you pre-implantation directly. They'll just give you the picture, and they'll ask you this, because I think I've seen a question like this, and the option should be PCR. Okay, next. What is the primary function of restriction endonucleases in molecular biology? And I think we know this. It's going to be degradation of foreign DNA. And I've told you guys how restriction endonucleases are like molecular scissors. They go and snip the DNA where we find the palindrome. So that's that. And it's an important concept, guys. Reverse transcriptase and restriction endonuclease. Okay, moving on. A patient is prescribed a new antiviral drug that functions by binding to the active site of a viral enzyme, preventing its normal function. The drug only becomes fully effective after the enzyme undergoes a conformational change upon binding. Which model of enzyme-substrate interaction does this drug's mechanism most likely reflect? So what do you guys think it would be? I'll tell you. It's going to be the induced fit hypothesis, because it says in the question itself that the enzyme is undergoing a conformational change. And when you see this conformational change in the question, it has to be induced fit hypothesis, because that's the whole concept of induced fit hypothesis. It's going to induce the fit. Even if it doesn't fit, it's going to induce it. And it's a key if you see the word conformation, just know that it's induced fit hypothesis. I don't think there's much to explain, except for I added this picture. You see that this is the substrate and this is the active site. You see the active site is a lot different than what the substrate looks like, but the substrate would still make it work. That means it's inducing the fit. So that means the enzyme is the one going the conformational change. And okay, moving on. Question 14. Alcohol dehydrogenase ADH requires oxidized nicotinamide adenine dinucleotide, which is NAD, for catalytic activity. In the reaction catalyzed by ADH, an alcohol is oxidized to an aldehyde as NAD is reduced to NADH and dissociates from the enzyme. The NAD plus is functioning as was it going to be a coenzyme, co-substrate, coenzyme prosthetic group, or cofactor? Okay. The answer is going to be B, coenzyme co-substrate. Well, it's because if you see in the question itself, it says that it's going to dissociate from the enzyme after doing its thing. So NAD is a coenzyme co-substrate. And also, I'll tell you why it's not coenzyme prosthetic group. It's coenzyme co-substrate because it's reducing to NADH. It's itself going under a change, and it's also helping the enzyme disrupted undergo their change. So it's a coenzyme co-substrate, and it's a coenzyme because it doesn't fully participate in the reaction. It's just going to help boost the reaction. So NAD plus transiently binds to the enzyme and leaves in a changed form, classifying it as a co-substrate as well as a coenzyme. So this is the reaction, what you see here, the ethanol turning to acetaldehyde. I think pretty explainable. Moving on. Okay, question 15. Which of the following statements best describes allosteric enzymes? They follow the Michaelis-Menten kinetics and display a hyperbolic relationship, or they catalyze only the final steps of a metabolic pathway, or they catalyze committed steps in metabolic pathways and display a sigmoidal substrate velocity curve, or they have a single active site and are regulated solely by substrate availability. The answer is C. They catalyze the committed steps and also display a sigmoidal substrate velocity curve, not a hyperbolic. And so basically, allosteric enzymes are those enzymes that catalyze the rate limiting step, or which is also called as the committed step. That means if that step occurs, the reaction and is bound to happen no matter what. If that step occurs, you will get the final product some or the other way. And they often have multiple subunits, not single, and they show a cooperative substrate binding, producing a sigmoid, like an S-shaped curve, not a hyperbolic. A hyperbola would be like this, and the sigmoid would be like this, if that makes sense. And moving on. A drug acts as a non-competitive inhibitor of an enzyme. Which of the following changes to Km and Bmax confirms this mechanism? So read it carefully. It says non-competitive inhibitor. So what do you think it's going to be? Km decreased, Vmax increased, or Km increased, Vmax decreased, or Km unchanged, Vmax decreased, or Km increased, Vmax unchanged? It's going to be C, Km unchanged and Vmax decreased. So first let's know what Vmax and Km is. So Vmax is the maximum velocity, or like the amount of substrate per unit time. And Km is an indicator of affinity of the substrate for its enzyme. So Km basically is the point where 50% of the enzyme is occupied by the substrate. And the explanation would be, so this is directly from your slides, the non-competitive inhibitors inhibit and bind to a domain other than the active site. So if you see in this picture, the inhibitors, the non-competitive inhibitors, they can bind to the enzyme directly, or they can bind to the enzyme substrate complex as well. And either way, there would be no production of the end product. And because they bind to the enzyme or the enzyme substrate complex, that means they're not reducing the substrate, or even if we add more substrate into the reaction, the inhibitor would still do its same. That's why the Km stays the same. It doesn't affect the enzyme's ability to bind to its original substrate. Therefore, Km stays the same. And Vmax decreases because it impairs catalytic function. As you see here, there's no production of the end product. That means it's impairing the catalytic function, therefore Vmax decreases. And Km stays the same. And this is important. You will have a question on either of these three. They will either give you competitive inhibitors, or non-competitive, or uncompetitive. And it's very, very important. And I feel like if you, oops, sorry. Okay, just trying to move this. Okay, so if you guys see a question on this, please remember that competitive increases Km and no change in Vmax, and non-competitive, no change in Km, an increase in Vmax, and uncompetitive decreases both Km and Vmax. And I added the explanations on this table. If you remember this table, you're good to go. Okay, next. A patient presents with signs of chronic liver disease. Serum enzyme tests show elevated AST and ALT. The AST is disproportionately higher than ALT. And which underlying condition is this pattern most suggestive of? A, viral hepatitis, B, fatty liver, C, cholestatic liver disease, or D, alcoholic liver disease, or E, hemolytic anemia. Now, I think by now you know that ALT and AST both are related to liver. But if you see the options, everything is about the liver. So how do we come to the conclusion? You see in the question itself, it says that the AST is disproportionately higher than ALT. This should point you to the fact that it's alcoholic liver disease. And I'll tell you why. After another question, I have an explanation of both of these questions together. But just remember that AST, if it says disproportionately higher than the ALT, or even if they give you numbers in the question and you see that the AST is higher, it has to be alcoholic liver disease. And next question. A 57-year-old man reports severe substernal chest pain 3 days ago. 3 days ago, remember. And which laboratory test is most appropriate to diagnose myocardial infarction? A, CKMB, or B, LD1, lactate dehydrogenase 1, or C, aspartate aminotransferase, or D, troponin I. It's going to be troponin I. Now, it's because you see in the question, it says 3 days ago. That should point you to troponin I, because troponin is the one that's elevated for up to 10 days after a myocardial infarction. Is the only one that's elevated up to 10 days. It's not going to be creatine kinase, because creatine kinase is elevated for only up to 2 days. I'll show you this part. So guys, I spent a lot of time on this, and if you remember this, you're good to go for these clinical enzyme lectures. You don't do the lecture, just do this slide and you're good to go for this, for any question based on this lecture, clinical enzymology, and expect to see at least 2 questions from this lecture. And liver and heart are the most high yield out of all of these. So let's start with liver. In the liver, if the AST is higher than the ALT, it has to be alcoholic liver disease, the question we just did. And if the AST is lower than the ALT, it could be a fatty liver or a liver damage. And if there's GGT, which is gamma glutamate something transferase, it's going to be a biliary obstruction, and it's checked to, or if it's checked to see if the ALP is of liver or bone origin. So ALP is also high in bone diseases, and if we find the ALP is high, we want to conclude if it's from the liver or if it's from the bone. So we would do the GGT, and if we find it, that means it's from the liver or it's a biliary obstruction. And then we have ALP, which is in cholestasis, which is basically another name for biliary obstruction, and or bone disease. Wait, I think cholestasis was gallbladder obstruction. Yeah. Okay, next is bone. The only enzyme for bone is ALP. So in the question, if you see that they're only talking about the ALP, it has to be something about the bone. It's osteomalacia, rickets, or Paget's disease. Paget's disease is something you'll take in second year when you're in MSI. So don't worry about it. If you see anything bone related, it's going. If you see ALP, it has to be something bone related. And either going to be either of these 3, and most probably they're going to ask you about osteomalacia or rickets. And then heart, very, very, very high yield, very high yield. And I'll tell you why, because there are 2 types of questions that come. If you see in your question that the patient is coming, like, after 3 days or like after a week, you're going to check for the patient's troponin. It either troponin I or troponin T. And it's the most specific and sensitive, and it's for the initial diagnosis, and it's elevated for up to 10 days. Therefore, even if the patient said, oh, they had like a myocardial infarction or like a substernal pain like 1 week ago, test for troponin, not for creatinine kinase. Creatinine kinase is elevated for only 2 days, and it's less specific, and it's mostly used to see if the patient is at the risk for getting a reinfarction. And the third one is myoglobin. Now, myoglobin is an early marker, and it's less specific. Like, it comes and goes. It's not elevated for quite long. And next is salivary glands. And the only enzyme you have here is the alpha amylase. So alpha amylase is in salivary gland disorders like mumps, or if there's salivary calculi, which is like obstruction. And then we have pancreas. They can't test you on pancreas, but I feel like the heart and the liver are the most important. And for pancreas, we have 2. We have alpha amylase, which is high in acute pancreatitis, but is not specific, because alpha amylase can also be elevated in salivary glands. And then we have lipase, which is the most specific for pancreas, and it's in acute pancreatitis. Okay, moving on. A 25-year-old healthy individual undergoes an overnight fast. Her fasting blood glucose is measured at 4 millimole per liter. 1 hour after consumption of carbohydrate-rich meal, her blood glucose rises to 8 millimole per liter. Which of the following tissues is least likely to show an increased rate of glucose metabolism in response to the rise in blood glucose? A. Pancreatic beta cells, B. Brain, C. Liver, D. Adipose tissue, or E. Skeletal muscle. So the answer is brain. Now, brain is the organ which requires a lot of energy and oxygen as well. So the patient is like, he was fasting. Of course, if he's fasting, the glycogen stores are going to get depleted, and we're not going to have enough glucose. So how does the brain work? It's going to it's not going to show an increased rate of glucose metabolism, because it actually cannot, I don't know how to explain. Because they use something called as GLUT1 and GLUT3, and whatever, and if you're in like a fasting state, your body is going to prioritize I think the brain the most. Wait, I'm getting confused. Oh, it says increased rate. Sorry, I thought it's going to show decline. Okay, so yeah, the brain is going to show an increased rate of glucose metabolism, because it's a very important organ and it needs glucose the most, and your body itself is going to prioritize the brain, and the glucose transport of the brain are GLUT1 and GLUT3, and they're highly prioritized. And also the other organs can use other things, and the other organs like the GLUT2 is in this is in like regulatory in liver and beta cells and kidney, and GLUT4 is like everywhere in your body except for the brain. And because the brain heavily relies on glucose, it has to be having a higher or increased rate of glucose metabolism in fasting states. Okay, moving on. Which of the following are primary stimuli of glucagon release? A. Decreased plasma glucose and increased hunger, or B. Decreased plasma glucose and amino acids, or C. Amino acids are released by muscle breakdown due to hunger, or D. Increased plasma glucose. Primary stimuli of glucagon release. It has to be a decrease in plasma glucose and amino acids derived from a meal. Now, this is because a decrease in plasma glucose is the primary glucagon release, because in overnight or prolonged fast, the elevated glucagon levels would prevent your body into going into a hypoglycemic shock. And amino acids derived from a meal contain protein that stimulate glucagon. So, I mean, if you see in the question, anything that says amino acids or protein-rich meal is going to stimulate your glucagon. Glucagon effectively prevents your body into going into hypoglycemia or hypoglycemic shock that would otherwise occur in as a result of the increased insulin secretion that also occurs after a protein meal. So after you eat food, your insulin and glucagon, both of them go up. Now, insulin goes up because it has to break down the glucose that you're getting from the meal. But if you secrete only insulin, your body is prone to go into a hypoglycemic shock. Therefore, your body would also secrete glucagon when you're secreting insulin after a meal. Suppose you had steak for dinner. Steak is protein rich, and you don't have carbs. You have only a steak. So what your body is going to do is it's going to release glucagon as well, because it doesn't want you to go into this hypoglycemic shock. So it's basically nature's way of saving you from a hypoglycemic shock if you had a steak instead of carbs for dinner. Okay, moving on. So Mohammed, a 30-year-old prisoner, has decided to protest by starving himself. He is surviving on water only and has not eaten a meal for more than 3 weeks. Under these conditions, what is the main source of fuel for Mohammed's brain? So it's going to be C, ketone bodies, because in prolonged fast or starvation, which is like more than a week or more than 3 to 5 days, the glycogen stores are going to get depleted, of course, and your liver shifts to something known as ketogenesis, which is producing ketone bodies from fatty acids. And the brain, which normally depends on glucose, it begins utilizing these ketone bodies instead as their major energy source to preserve muscle protein by reducing gluconeogenesis from amino acids. Okay, moving on. Why can't the liver utilize ketone bodies? So this is a good question, because the liver is the site of production of ketone bodies, but why can't the liver utilize these ketone bodies? Let's look at the options. So A is it lacks mitochondria, or B, it lacks enzyme theophorase, or C, it has insufficient ATP, or D, it lacks enzymes for ketone body synthesis. So the answer is it lacks the enzyme theophorase succinyl-CoA, or acetoacetate-CoA transferase. So it's the liver is the site of production of the ketone bodies during fast or uncontrolled diabetes, like in type 1 diabetics. And however, the liver cannot utilize its own ketone bodies because it lacks this enzyme called theophorase, and it is an essential enzyme that converts your ketone bodies into usable forms of energy. Now, this ensures the ketone body, and why this happens is because after your liver produces these ketone bodies, it has to send it to other parts of your body, like your brain and your muscles and your heart. Now, suppose the liver was able to use its own ketone bodies, I feel like the liver would be selfish and use everything for itself. Therefore, to prevent this from happening, your liver does not have something called as a theophorase, which is essential for converting these ketone bodies into usable forms of energy. Therefore, to remove it from the liver and send it out to heart, brain, and muscles and stuff, it's necessary. Okay, moving on. A physician is treating a type 1 diabetic patient who has neglected to take his insulin for 5 days. The patient demonstrates elevated blood glucose and ketone body levels. Now, ketone bodies are elevated due to which of the following? Elevated glucose levels, reduced B1, decreased fatty acid release from the adipocyte, or inhibition of liver oxidation of ketone bodies, or E, reduced muscle utilization. And the answer is elevated glucose levels. Now, it's elevated glucose levels because type 1 diabetics, they don't have insulin at all. They don't produce insulin at all because, yeah, they just don't do it. And because they lack, or more like an absence of insulin, it leads to uninhibited lipolysis. That means they are breaking down their lipids, and therefore, because they're breaking down their lipids, they have a lot of free fatty acids. And then the liver is going to take these free fatty acids into acetyl-CoA, and it's going to do ketogenesis. And it gets shunted into ketogenesis. That means it's going to bring it into ketogenesis. And ketogenesis is the production of ketone bodies, and therefore they have high ketone bodies. And this causes something known as DKA, or diabetic ketoacidosis. So remember, in type 1 diabetes, this very specific type of diabetes, they have something known as diabetic ketoacidosis, and they have like a fruity breath because of these because of the ketoacidosis. And next, Ashaz, you want to take over from
here? Yeah. Can you give me access to your screen?
Did you request access?
I'll just share it from my screen.
Oh, you'll see. Okay, I'm going to stop sharing.
Yeah.
Okay, go ahead.
What's this? Is everything visible?
Yeah, we can see.
So I'll be taking over from here. And yeah, let's start. A physician is treating a type 1 diabetic patient who has neglected to
Ashaz, I
take.
just did this question.
Oh, you just did it?
Yeah. You can start from the question 24.
Yeah, my
Okay.
bad.
Thank you.
Yeah. In type 2 diabetes, due to some residual pancreatic function, which symptom commonly associated with type 1 diabetes is typically absent. The answer would be C, ketoacidosis. As my colleague just mentioned about ketoacidosis, you can see how two questions were like two concepts, or this question was repeated many times, which means it's a really important concept. So you have a difference between type 1 and type 2 diabetes, okay? Type 2 diabetes is where your body increases insulin, and type 1 diabetes is your body not being able to produce insulin at all, okay? So in type 2 diabetes, it increases first and then it goes down because of insulin resistance. Type 1, insulin is being completely inhibited because type 1 diabetes is an autoimmune disorder which attacks your pancreas, right? But in type 2, you'll still have some insulin being produced. And this amount of insulin is often enough to prevent diabetic ketoacidosis, right? Remember, type 1 diabetes leads to a decreased insulin. And if insulin decreases, glucagon increases. This will cause increase in free fatty acids, which will be converted into ketones, increasing the amount of ketones in blood and causing diabetic ketoacidosis, which is a medical emergency. Okay. Now, which enzyme catalyzes the committed step of fatty acid synthesis and is regulated by both allosteric and hormonal mechanisms? Really, really important question. Okay, give it 2 seconds. The answer would be acetyl-CoA carboxylase. Now focus with me. Okay, this image right here, keep it in your head. 100% you're gonna get not one but two questions from this. Okay, imprint this diagram in your head and know that acetyl-CoA carboxylase is activated by two things, citrate and insulin, and is inhibited by palmitoyl-CoA and glucagon. I'll tell you each one of them, okay? The question, it said allosteric and hormonal mechanisms. Remember that citrate is an allosteric mechanism. Remember, the citrate is an allosteric mechanism. This was asked before in our exam, I believe. And insulin is a hormonal mechanism. Now, inhibition is by palmitoyl-CoA and glucagon. Inhibition by palmitoyl-CoA is because of regulation or feedback inhibition, because palmitate is formed, so the process of producing fatty acids will be a bit less now. And glucagon, because it works opposite to insulin. Right. Now, a patient with type 2 diabetes, hyperinsulinemia, his insulin is raised. Which one of the following describes the insulin's effect on fatty acid synthesis? We just discussed this, right? So you can see how important this concept is. This has been repeated many times in previous years, right? So the answer would be, yeah, insulin works by increasing ACC activity. Now, how does it increase it? Dephosphorylation. Glucagon does the exact opposite. It phosphorylates, right? Citrate dephosphorylates ACC, activates it, and more melanin-CoA and more free fatty acids. Glucagon phosphorylates. Remember, one more time. Um, Insulin dephosphorylates and acetyl-CoA carboxylase activates. Glucagon phosphorylates, the enzyme is inhibited. Okay? Remember this. Now, what are the first steps and the rate limiting steps in ketone body formation? Give you like 2 seconds to read this question. The answer is B. So first thing, your acetoacetyl-CoA will form, and the rate limiting step is at HMG-CoA synthase. I'll briefly go through the whole pathway because you have to remember this pathway. You have to. So first, in ketone body formation, we just discussed like 2 questions before, like how fatty acids convert to ketones, right? So this is how they do that. So you have your fatty acids, one fatty acid molecule and two acetyl-CoA molecules will first combine to form the acetoacetyl-CoA. Now this step is the first step of ketone body production. From here, you have the HMG-CoA synthase forming the HMG-CoA itself. Now this thing breaks down into acetyl-CoA and acetoacetate again, right? And yeah, it just goes back. So you have acetoacetyl-CoA with the help of HMG-CoA synthase, you're gonna have HMG-CoA, okay? Again, fatty acid plus 2-acetyl-CoA combine to form the acetoacetyl-CoA. From there, with the help of HMG-CoA synthase, they're gonna form HMG-CoA. So remember, the rate limiting step of ketone body formation is at HMG-CoA synthase. That's the enzyme. Okay. Now this breaks down into acetyl-CoA and acetoacetate, right? And the acetoacetate again breaks down into acetone and 3-hydroxybutyrate. These are the end products. And acetone and the 3-hydroxybutyrate, these are the ketone bodies which we find, right? Now statins lower cholesterol by inhibiting which enzyme? It's easy. This is asked again and again and again. I think this was asked like pretty sure just in the past 2 years, this was repeated, right? So the same concept. The answer, HMG-CoA reductase, the rate limiting step, right? Keep in mind the pathway of ketone synthesis we just discussed. So the rate limiting step, that's the one which is what do you call inhibited by statins. Now we have this thing. How does a PCSK9 inhibitor lower LDL cholesterol? So just by the question, you can know that PCSK9 is something which inhibits blood LDL cholesterol, right? Or which increases, sorry, which increases blood LDL cholesterol. So you inhibit PCSK9 to decrease it. Now, how does it do it? So the answer would be by blocking PCSK9 from degrading LDL receptors. I mean, that was common sense. But I'll explain the whole pathway to you. It's important for you to know that if you have PCSK9, it's going to increase LDL cholesterol, which is bad for you. So you have to inhibit PCSK9, right? Now, how does it work? So the liver cell on its surface, there's the liver cell on the surface, you're going to have a receptor for LDLs, which means these receptors are going to take up LDLs and bring it inside the cell. But these receptors, when they bring the LDL inside the cell, they will come inside too. The receptors will come inside, right? So they will have to come back. Okay. Now what happens is PCSK9 prevents the receptor from coming back up. So more of the LDL will not come in the liver cell and it's just gonna float around in the blood. And if there is LDL in the blood, it's gonna be bad for you, right? So just remember, PCSK9 basically inhibits reemergence of receptor or receptor resurfacing, right? That's what it inhibits, and that's why it's bad. So now question 30, this image right here, remember it. Please remember it. Okay? A 50-year-old man wants to naturally improve his heart health by raising his good cholesterol levels. So, which of the following lipoproteins must be increased? Remember, there are certain keywords which I'll be discussing an explanation for this one. Okay? There is bad cholesterol, there is good cholesterol, and then there's dietary cholesterol. I'll discuss all that. But for this one, the answer would be E. Okay? Why? First of all, this table right here, remember it. Okay? I think we got a question from this a lot in TBLs and I think even in the finals, right? That's what I remember. Um, yeah, there's an almost guaranteed question. Know what the different types of lipoproteins are in this image. So you have lipoprotein A, which is the least dense. The density is less than 0.93. A is chylomicron. B is VLDL. C is, I believe, LDL. D is MDL or medium density lipoproteins, and E is high density lipoproteins, right? So high density lipoproteins are usually the good cholesterol. You have LDL, LDL is bad, HDL is good. Remember this. LDL bad, HDL good. So if you have a question which focuses on heart health and asks you which one should increase, HDL. Okay. If someone's eating junk food, which one would increase? LDL. Okay. Or if someone is eating any food, first thing chylomicrons will increase. Okay. They'll go into the liver and then they will convert HDL or LDL from there. Which lipoprotein transports dietary triglycerides? I repeated this like 2 times or 3 times before. Right? It's really easy. The answer is chylomicrons. They originate from the intestine. Okay, so now for the pathway of chylomicrons, you have to remember the pathway of chylomicrons, right? Um, so first of all, you're gonna have fat in your intestines from the food you ate, and this fat is gonna be emulsified to form something called micelles, right? The micelles are going to be taken up as chylomicrons from the epithelial cells, and these will drain into the lymphatic system. From the lymphatic system at the thoracic duct, which is the most important duct in all of the lymphatic system, right? So from the thoracic duct, it's gonna reach blood vessels, okay? And from the blood, it's gonna reach the peripheral tissues. So once it reaches the peripheral tissues, the chylomicron is basically this thing, the casing. You could imagine it as a casing with triglycerides in it or fat in it, right? So now you have to open the case. How are you going to do it? By something called lipoprotein lipase. Really, really important. This name, lipoprotein lipase, really important. Remember it. Okay. So these are now broken down with the lipoprotein lipase. You'll have what's left. Okay. The main thing is taken out, what we need. Now, what's left is something called chylomicron remnants. Now, this will be taken to the liver, right? So, yeah, that's it. Now, you'll have a lot of keywords. Okay, this is also a really repeated concept. Okay, 3-month-old infant brings their child to the ER. Okay, they are disturbed by the extreme lethargy and near comatose state of the baby. So the baby is also hypoglycemic, no ketone bodies, and elevated C8 to C10. These three: hypoglycemic infant with no ketone bodies and elevated fatty acids, which are of medium length, a deficiency of which enzyme is most likely responsible for this presentation. Now, this is a pretty easy answer. Okay, it's really repeated, it's really easy. The answer is medium chain acyl CoA dehydrogenases. Now, I'll explain something. Loki, the questions from this concept are not really complex. You just have to know 3 symptoms, 3 keywords from the questions. If you see these keywords, then you should be fine. The 3 keywords are hypoglycemia, low ketones, and if you see C8 to C10, medium chain fatty acid increase. These three, hypoglycemia, low ketone, and C8 to C10 fatty acid increase. If you see these three, it means it's an MCAT deficiency. That's it. The questions are not going to be really complicated. You will have a question on this, but it's not going to be complicated. So don't overthink it. Okay. But for the processing of it, we just talked about fatty acid synthesis. So how it goes to all the way to 16 carbons, right? Now during oxidation, it's gonna eventually break down. So it's gonna go from a 16 carbon molecule fatty acid to a 14 carbon to 13 to a 12 to release, to keep on releasing acetyl CoA. And this acetyl CoA is used for other processes to provide us with energy, right? So once we keep on releasing acetyl CoA, once we reach at the medium length, people who are normal, they're gonna have the enzyme called MCAD. That's the enzyme, okay? But people who don't have this enzyme will not be able to break down fat past this level. Yani C16 all the way to C10 or C8. After that, it's not breaking down. Okay? Because typically normal people should be able to break it down all the way to its last carbon, right? That's not happening here because of this enzyme being deficient. So now what's gonna happen is you're gonna have accumulations of medium chain fatty acids, and the fat is not gonna be used efficiently for energy. Okay, that's why these people cannot fast because they need a constant supply of fatty acids because they cannot use all the 16 carbons present in a normal chain. So now to compensate for it, they need more fatty acids to use the carbon they need because their carbon requirements are just as much as us. Okay. So that's why in infants, an overnight episode might occur if they don't eat anything, right? Now, which of the following represents a negative nitrogen balance? Remember the concept. What is a positive nitrogen balance? What is a negative nitrogen balance? Remember it. Okay. So actually, we have two answers here. Okay. This confused us. I know this confused us, but there are two answers. Now, for the first one, the guy who has recovered from extensive burns, he had burns, right? See, first of all, let me explain what positive and negative nitrogen balance is. It's a really important concept. You will get a question on this. Okay. So, negative nitrogen balance, loss of nitrogen is more than the gain of nitrogen. Okay. Basically, it's a catabolic state. For example, first of all, when we are talking about nitrogen, it's mostly about amino acids. Amino acids have a nitrate group in them. So that's the nitrogen we have in our body, right? So if you have an excess of nitrogen, which means you're either healing, new tissue is forming, or you're building muscle. Something of an anabolic state. You're building up something. Okay, you have more of these ingredients to build up, so you're gonna build up, right? But if you have less of the ingredients, so the body is gonna use what you have already. Okay, so that's gonna be a catabolic state. So positive nitrogen balance is when you have excess amino acids, excess nitrogen, and you use that to build yourself, anabolism. Negative is when you have less than what you need, okay, which is catabolism. The patient had burns, okay, and that burns caused tissue loss. So you're losing tissue here, negative balance. Patient has kidney failure and the renal tissue got destroyed because he has renal failure, right? And these people who are chronically sick are usually in a catabolic state. So you could take that hint too, and it's going to be nitrogen or negative nitrogen balance again, right? So now, 5-day-old neonate is brought into the ER with lethargy, vomiting, and hyperammonemia. Okay, there's elevated orotic acid. Which enzyme deficiency is most likely responsible for this condition? Is it arginosuccinate synthase, OTC, arginase, carbamyl phosphatase synthase 1, and glutamate dehydrogenase? So the answer is B. Okay, you're right. The answer is B. Now, this image right here, really important. I believe the image in the lecture is also pretty simplified image, but if you guys don't understand it, I think this should be a good image. So I won't really go into the details for time's sake, but Um, First of all, you're gonna have carbon dioxide and you're gonna have ammonia. Okay, with the help of carbonyl phosphorus, you're gonna have carbonyl phosphate, and this plus L-ornithine. When you use OTC, you're gonna have citrulline. Okay, this is the step you have to know for this, and usually questions are asked on this step right here. Okay, you have to know these two, the other steps, but most of the questions which are asked are usually from this step, right? So for OTC deficiency, when you read the question, okay, you're gonna see two symptoms: hyperammonemia, not hyperammonia, hyperammonemia. Hyper is increased, ammonia, emia in blood. So you have an increased ammonia in blood, and most importantly, you have orotic aciduria. Okay, so hyperammonemia, orotic aciduria. When you see these two, the answer is OTC deficiency. No questions asked. Okay. Now, a newborn with sweet smelling urine, ketoacidosis, likely has a defect in which enzyme? Okay. The answer is branch chain ketoacid dehydrogenase. It's the questions from these are going to be really simple. You have some buzzwords, just keep that in mind. Okay. So, yeah, this is a highest yield question. You're going to have maple syrup urine disease. The typical symptoms you're going to see here are going to be sweet smelling urine and ketoacidosis. Okay. What happens here in this disease is when you have a keto acid, which is of a branch chain, right? These keto acids are not going to be broken down. Okay. Like leucine, isoleucine, and valine. These are branched in their structure, right? So to break these down, you need different enzymes. The enzymes here are branch chain alpha keto acid dehydrogenase. If you don't have these enzymes, you're going to have accumulation of branch chain amino acids, and this will increase what do you call ketoacidosis. This will cause ketoacidosis because these are keto acids. Okay, and this will cause sweet smelling urine and ketoacidosis. Remember, sweet smelling urine, ketoacidosis equals to maple syrup urine disease. If you want to remember it, you know how syrup is sweet, right? So that's why it's actually named sweet smelling urine. Okay. You're counseling a mother whose newborn just screened positive for a deficiency of phenylalanine hydroxylase enzyme. You inform her that her child will require dietary supplementation of which of the following? Think of the pathways we used here. Think of the pathways. Okay. This is from tyrosine metabolism. Phenylalanine converts to tyrosine and it uses an enzyme. Okay. If you answer tyrosine, it's correct. Why? Phenylalanine converts to tyrosine with the help of an enzyme called phenylalanine hydroxylase. Right. So this tyrosine is required for a lot of stuff. It's used for melanin production. It's used for hormonal production and all those. Right. Now, if you don't have enough tyrosine in your body, you're gonna have many other issues. So now, mm, if the person does not have the enzyme, they cannot convert phenylalanine to tyrosine. So you have to supplement them with tyrosine to maintain normal function. So that's the whole concept of this thing, right? Remember, if you have PKU, um, if there is a phenylalanine hydroxylase deficiency, if there is PKU, phenylketonuria, you give tyrosine as a supplement and limit phenylalanine. Okay. Which disorder is caused by defective tyrosine metabolism? Give me a second.
Recording in progress.
So the answer here is going to be albinism. Okay. Now, focus with me. Tyrosine metabolism, you're going to have three diseases. you have albinism, you have phenylketonuria, and you have alkaptonuria. And all three of these diseases are important. Okay, first of all, I'll start with albinism because that's what the question was on. Okay, so albinism, you're gonna have a lack of melanin because I told you tyrosine converts to melanin. Okay, so if what do you call that process does not happen, you're gonna have albinism. Now, the second problem in tyrosine metabolism is phenylketonuria. This is where there are problems in the production of tyrosine, right? We did this question like two questions before. If you have productions of tyrosine, they have their own bunch of problems, okay? And then you're gonna have alkaptonuria. There is this enzyme called homogentisate oxidase, which breaks down tyrosine. So tyrosine production is normal, but then you have to break it down. And if that does not happen, tyrosine accumulates, and you have something called alkaptonuria. Okay, an enzyme which is deficient here is homogentisate oxidase. So just remember one more time, three problems in tyrosine metabolism: albinism, PKU, and alkaptonuria. Okay. So a 42-year-old woman who has been taking medication for high blood pressure visits her doctor for routine checkup. During the examination, the doctor notes that her creatinine levels are elevated, which indicates impaired kidney function. Creatinine is produced by which of the following precursors? So creatinine comes from what do you call creatine, and creatine is produced by some precursors. Think about that. Think about the three precursors which it uses. Okay. So the answer is glycine, arginine, and SAM. So, S-adenosylmethionine. That's the full form of this, but just call it SAM. Okay. Again, as I said, creatinine is a breakdown product of creatine, and creatine comes from glycine, arginine, and S-adenosylmethionine, or in short, SAM, which is a methyl group 12. Again, creatine comes from glycine, arginine, and S-adenosylmethionine. Okay, this concept is repeated a lot, so just keep it in mind. Glycine, arginine, and SAM. Okay. Now, a 7-year-old boy is brought to the clinic by his parents due to progressive visual problems and frequent falls. Visual problems, frequent falls. On exam, he is tall and thin with long limbs, mild intellectual disability, and his lens is displaced inferonasally. You have some hints here. Tall and thin, intellectual disability, and the displacement on the lens, and it's inferior nasal. Inferior down, nasal towards the nose. Okay, so basically looking at the medial cancers, right? He also has an early onset of thromboembolic events. Okay, there are elevated levels of homocysteine and methionine. Okay, so which of the following enzyme deficiencies is most likely the cause of this patient's condition? Okay, run the whole thing. Homocysteine is increased, methionine increased. So which one will it be? Homogentisate oxidase. Okay, now I'll tell you how. First of all, when the child has homocystinuria. Okay, now if you see symptoms like tall and thin, that's one thing. It's like Marfan syndrome, but it's not really Marfan syndrome. People with Marfan syndrome, they do not have intellectual disability. That's one thing. People with homocystinuria have intellectual disability. Their body shape will be similar to someone who has Marfan syndrome, which are they're like tall and thin. Okay, and the lens dislocation. This is the biggest differentiator between Marfan syndrome and homocystinuria. Okay, in Marfan syndrome, the lens of the eye will be dislocated up and outwards in this direction. In homocystinuria, it's going to be displaced down and inwards, exactly opposite. Okay, and then you're going to have thromboembolic risk. Okay, and elevated homocysteine and methionine. Okay, now uh, a 60-year-old woman presents with weakness, fatigue, nausea, and diarrhea. Her medical history reveals long-term severe rheumatoid arthritis treated with regular methotrexate. Okay, which of the following condition is most likely a complication arising from her methotrexate treatment? Methotrexate is a really important drug which is used in rheumatoid arthritis, but it has a really specific side effect which many other drugs don't have. So please remember this, okay? Now the answer is going to be folate deficiency. Why is it? That's the whole deficiency. That's the whole problem with methotrexate. Okay, so we need tetrahydrofolate for DNA synthesis, right? What happens is folate converts to tetrahydrofolate by something called dihydrofolate reductase. That's the enzyme which converts folate to tetrahydrofolate, and THF will be used for DNA synthesis. Now, if you inhibit the enzyme, you won't have tetrahydrofolate, and DNA synthesis will be impaired. Okay, methotrexate inhibits the enzyme. So that's all really you have to know, but it's an important thing to know. A child with self-mutilation, neurological impairment. Easy question. Repeats every single exam. Okay. AGPRT, Lesch-Nyhan syndrome, is a purine salvage disorder. Really high yield. The enzyme involved is HGPRT, which is short for hypoxanthine guanine phosphoribosyl transferase. H hypoxanthine, G guanine, P phosphor, R ribosyl, T transferase. Okay. Now the important symptoms, these I took this from your slides directly. Okay. You're gonna have hyperuricemia, HGPRT, hyperuricemia, G is good, P is pissed off. So the patient is gonna be really angry all the time and agitated. R is retardation. Retardation is not really a right word to use. Now, we won't get into it, okay, but they'll have intellectual disability. And T is for hypotonia, okay. Now, question number 42. A 6-month-old infant is brought into the clinic due to failure to thrive, developmental delay, and frequent infections. Failure to thrive, developmental delay, and frequent infections. An examination, the child appears pale, okay, and the patient has megaloblastic anemia that does not improve with supplementation. Biggest thing. Megaloblastic anemia does not improve with vitamin B12 or folate supplementation. Okay. Urine analysis reveals elevated levels of orotic acid. Okay. This is a trick question. Stay with me. This is a trick question. Which of the following is most likely underlying enzyme deficiency? Okay. Now, many of y'all will read the orotic acid and will choose OTC. You will be wrong. Okay. Remember, I told you hyperammonemia and OTC or orotic aciduria. Okay. Hyperammonemia and orotic aciduria. These two are responsible, or these two are seen in OTC deficiency. But here there is no case of hyperammonemia. Instead, there is megaloblastic anemia that does not improve with vitamin B12. Right. So the answer would be UMP synthase. Now, again, remember the key differences here. In UMP synthase deficiency, you have megaloblastic anemia, which does not respond to supplementation, and you have orotic aciduria. In OTC enzyme deficiency, you're going to have hyperammonemia and orotic aciduria. So don't get confused between them. Okay? Read the question properly. Remember this. Now, I think this is the last question. Allopurinol treats GERD by inhibiting which reaction? Really easy. Okay? The answer is xanthine to uric acid. Now, I'll briefly go through this diagram here because you have to know this diagram, by the way. Okay. You have the nucleic acid will break down to purines, and the purines will break down to hypoxanthine. Okay. Xanthine oxidase catalyzes two steps: conversion of hypoxanthine to xanthine, and from xanthine to uric acid. Hypoxanthine to xanthine, xanthine to uric acid. Both of them use xanthine oxidase. Now, from uric acid, you're going to have something called allantoin formation. Allantoin is usually seen in animals, I believe. We don't have the process. We just release uric acid through urine. Okay. Now, what happens in GERD is that we have an increased level of uric acid in the blood and it accumulates in our joints. So we want to decrease uric acid levels. Okay. We can do that in two ways, either by decreasing production or increasing excretion. Okay. So if you want to decrease the production, you're gonna stop the formation, or like you're gonna stop the action of xanthine oxidase. So it's never gonna become uric acid, it stays at xanthine. Okay. So the most important drug in this case is allopurinol. Remember that drug's name, allopurinol. Okay, okay. So that's uric acid. Like what do you call? That's stopping the production of uric acid. Again, inhibit xanthine oxidase, stop the production of uric acid, and stop its accumulation in the blood. Now, other thing is you can increase the outflow of it. So if you use something called probenecid or sulfenpyrazone, these promote excretion of uric acid. Okay, that's all you need to. And yeah, that's it.
Congratulations to everyone finishing MOL. And congratulations on finishing first year. Please give us a good feedback. That's it, right?
Yeah, I think that's it.
Okay. Thank you all. Bye.